Good day to all the assessor. My name is Win Win. The topic of my presentation is the role of bioinformatics in microbiome and metagenomic studies. And these are my content. Microbes are everywhere. Why do we say so? Because estimation number of prokaryotes on Earth contributes 5 million trillions, which account almost half of the biomass on Earth. And interesting thing is they can be found in even extreme environment, such as North South Poles, volcanoes, the ocean trenches, or upper atmosphere. And all plants and animals are actually very closely related to all these microbial communities, as they make necessary nutrients, metals, and vitamins available to their hosts. I would like to start with definitions. There are two terms, microbiota and microbiome. Some may mistakenly use both terms interchangeably, but they actually don't mean entirely the same thing. I'll explain further. Okay, microbiota actually only means the microorganism, which includes like archaea, fungus, or bacteria. Okay, whereby microbiome actually, besides the microorganism that inhabit the specific site, it also includes the genome and the surrounding environmental conditions of the microbiota. So in other words, it's a microbiome. It's a community of microbiota that live in a particular environment. To understand what is metagenome, it's the collection of all genomes of the microbiota, which is obtained using metagenomic sequencing method. And then the study of metagenome is known as metagenomic. It is now the practice of metagenomics to study the microbiome population, structure, and function. There are two types of metagenomics. First one being targeted metagenomics, also known as amplicon-based sequencing. And the second one, shotgun metagenomics, also known as whole genome shotgun. So as the name implied, for targeted metagenomics, it sequences specific genetic markers or conserved region within the microbial communities. So as you see from the figure, targeted, there is a few types, 16S, 18S. So 16S is for the bacteria, and 18S is, or the ITS, full terms means internal transcript spacer regions, are for the fungi. So these regions have variable sequences that are useful for the identification of different organisms present in the sample. However, with this method, identifying the organism at the species level can be challenging, as this method generally does not provide strain level resolution. But for shotgun metagenomics, unlike the targeted sequence methods that focus only on a specific genetic markers, shotgun's metagenomics sequence all the DNA which is present in a sample. So they do random sequencing of all genetic material in the samples. This actually provides detailed information on both the taxonomic and functional compositions of the microbial community, and it also allows species level identifications. This picture shows the general workflow for metagenomic studies, which begins with sample collection and then extracting the DNA to prepare the DNA library, followed by sequencing those DNA and reconstruct the sequences. Okay, contigs, as you see from the pictures, are overlapping regions from different short reads. After that, those genome sequence will be assembled. And finally, tools and software will be used for further analysis. As I've presented on what is metagenomics, the types, and its workflow, now let me introduce what is bioinformatics and why is it so important for bioinformatics to come in metagenomic research. Bioinformatics is an interdisciplinary field that merges biology and computer science, mathematics, and IT to manage and analyze huge biological data. Bioinformatics focuses on the development of databases and algorithms designed to enhance and speed up biological research, including metagenomics. It creates efficient methods for data retrieval and analysis, ultimately to help in resolving complex biological questions. Now we go into bioinformatic tools used in those workflow process. So tools like FastQC and Trimomatic actually helps to for better sequence quality control and pre-processing, as they remove low quality reads and contaminants, which minimize the artifacts and support downstream analysis. So they also trims by trimming the adapters, filtering, and decontamination. So FastQC is a tool where provide a simple way to do some quality control check on raw sequence data coming from high throughput sequencing pipelines. It provides a modular set of analysis which you can use to give a quick impression of whether your data has any problems of which you should be aware before doing any further analysis. Trimomatic is a bioinformatics command line tool designed to pre-process Illumina FastQ sequencing data. So it performs a variety of useful trimming tasks for Illumina pair-end and single-ended data. The selection of trimming steps and their associated parameters are supplied on the command line. Amplicon taxonomic profiling is a step to identify which microbes are present, either bacteria, virus, or fungi from the sample. The famous one being QME2, Data2 is for 16S Amplicon, and MGRAS for shotgun profiling, depending on what's the goal of the analysis. And Data2 is a standard denoising approach for high resolution Amplicon data, and MGRAS actually can provide rapid community profiling and annotation. It uses reference-based tools such as the Kraken2, Meta5N3, Kaju to classify sequence by matching to known microbiome genomes. The short read sequence need to be assembled and bind to reconstruct the genomes and strains, enabling linking genes to taxa. Okay, so here there are a few genome-centric tools such as the Rosella and the Aviary. They actually assemble contigs and recover metagenome assembled genomes to improve recovery from complex samples. And another one, MegaHit, is also an ultra-fast and memory-efficient NGS assembler. It is optimized for use in metagenomes. As mentioned, binding, they group contigs into genomes and resolve strain diversity. Tools like LoRiKit support higher resolution population analysis. Functional mapping and annotation tools such as PROCA, KGG Mapper helps to predict the metabolic function and pathway of the microbial community by mapping genes or pathways from the sequence and integrate multiple reference databases, which provide streamlined functional annotation. So in other words, it assigns genes to known metabolic pathways from the database and help infers ecosystem roles such as how microbes contribute to digestion, soil fertility, or disease. Verbox is a tool which offers an integrated phage analysis web server for particularly phage or viruses. So they help to identify virus, predict lifestyle and host, or even do gene annotation, or even provides functions to analyze the phylogenetic tree. Now, with the advancement of various bioinformatics software available to help researchers to analyze and interpret the complex microbiome data gathered by metagenomics, it assists in predicting the diversity of microbiome as well as identifying new species, studying the role of microbial function, and understanding the interactions among microbial communities. By studying the genetic materials of this microbiome, scientists can now begin to understand these microbes at a very fundamental level and isolate the genes that give them unique abilities to survive under extreme conditions. Besides, metagenomics and NGS is now widely used even for epidemiological surveillance from a One Health perspective to broaden the surveillance and characteristic of existing pathogens or new ones in humans and animals. It also provides pivotal discoveries in the field of medicine, such as association between microbiome and diseases such as metabolic, digestive, and cardiovascular diseases. Having complete genome sequences and their potential to provide a greater insight into the microbiome world and its capacity could have very far-reaching implication even for environment, health, energy, and industrial application. So I list a few genome applications here, including waste cleanup. Deinococcus radiodurans is a world toughest bacteria as it has the most radiation resistance organism known. Scientists are very interested in this organism because it has a potential usefulness in cleaning up waste site that contains radiation and toxic chemicals. Besides, climate change studies also uses microbiome because in order to reduce the atmospheric carbon dioxide level, one method is to study the genome of microbes that use carbon dioxide as their sole carbon source. And another application includes alternative energy sources. Scientists are now studying the genome of Coriobium tepidum, which has an unusual capacity for generating energy from light. Now with bioinformatics to discover microbial genomes, to analyze microbial genomes, scientists has also managed to examine the virulence regions from a number of antibiotic resistant genes that may contribute to the bacterium's transformation from a harmless gut bacteria to a terrible invaders. So by discovery of the region known as the pathogenicity island, this provides useful markers for detecting pathogenic strains and help to establish control to prevent the spread of infection and antibiotic resistance. Besides, there's a gene from Bacillus thuringiensis controls also a number of serious pests that has been successfully transferred to cotton, maize, and potatoes. This new ability of the plants to resist insect attack means that the amount of insecticides can be reused and can be reduced, and hence the nutritional quality of the crops also can be increased. Metagenomic is now a very promising approach for investigating the biodiversity of microbial community and also contribute to the gene pool of the environment, as the direct sequencing of the DNA does not need any prior knowledge of the gene sequence. It is now possible to assess the sequence of genes and thereby determine the function of these genes. So, but one thing to take note of that metagenomics is highly dependent of the DNA sequence given. So the quality control of sequencing is actually very important to ensure accurate analysis. So that's how bioinformatics come into the picture. Besides, the sequencing also release massive amount of data. So employment of the bioinformatic program is crucial for this analysis and the storing of this massive amount of data as well. So with bioinformatics now, in metagenomics, it actually speed up the process and ensure accurate and reproducible results to create a positive impact to mankind and our environment. One extra thing to take note is although there are many software applicable to analyze metagenomic data, what is important option to choose the superiority of a software over the others is the analysis speed, the qualification of the analysis process, and the ability to link the different gene databases, and the user friendly of the software. These are my references. Thank you.